So we have the phospholipid bilayer with the hydrophilic heads and the hydrophobic tails. And then we have membrane proteins that have various functions. There's a lot of different ones. There's 6 different types listed here. There are receptor proteins, channel proteins, carrier proteins. There are proteins that act as enzymes. They're no different from any other enzyme except they're attached to the plasma membrane. There are anchor proteins and then proteins that act as identifiers. So this table lists the membrane proteins and it gives you the function of each one and an example of each one. So for receptor proteins are sensitive to specific extracellular materials that bind to them and trigger a change in the cell's activity. For example, there's a receptor for insulin on certain plasma membranes, like plasma membranes of liver cells and plasma membranes of muscle cells. They'll have a receptor for insulin on their plasma membrane. The insulin will bind to the receptor, and that will allow the cell to bring glucose into the cell and lower the blood glucose levels. Channel proteins form a channel that ions and other materials can pass through. Carrier proteins are similar to channel proteins, except the substance that's being transported actually chemically will bind to the carrier protein, and the carrier protein will then change shape, and it will either it will bring the whatever the substance is, it will bring it across the plasma membrane. Then we have enzymes that speed up chemical reactions. We have anchoring proteins. The example here is inside the cell, anchor proteins bind to the cytoskeleton. Outside the cell, anchor proteins attach the cell to extracellular protein fibers or to another cell. Then we have recognition proteins or identifiers, and these are going to be ones that are used, for example, in tissue compatibility, determining if someone's tissue is compatible to so that they can be a donor for another person. These recognition proteins that function in tissue compatibility are called the MHC proteins, and that stands for Major Histocompatibility Complex, and histo means tissue. So really histocompatibility means tissue compatibility. Okay, so we have these types of membrane proteins, and then we have membrane carbohydrates that are either attached to phospholipids, and we call those glycolipids, or they're attached to proteins and they're called glycoproteins. In fact, the recognition proteins are usually glycoproteins. Which functional class of membrane protein allows water and small ions to cross the plasma membrane? Well, it's going to be channel proteins, and we could also be talking about carrier proteins, but the carrier proteins would chemically bind with the ions or with water. So I think here, I'm sure that it means channel proteins here. Channel protein. Okay. Then we have the permeability, the ability of the plasma membrane to allow substances to pass. With regard to permeability, we call the plasma membrane selectively permeable. It's a selectively permeable membrane. That means some things can cross, but others can't. Okay, when we describe movement across the plasma membrane, we describe it as passive or active. Passive means no energy is required. Examples of passive processes are diffusion, osmosis, and facilitated diffusion. And active processes do require energy. Typically, that energy is from ATP. Examples are active transport and vesicular transport. And carrier-mediated transport, in other words, transport involving a carrier protein, can be active or passive. So let's go through all the different examples. The first example is an example of passive transport, and it's just diffusion. Diffusion is defined as movement of substances from areas of high concentration to areas of low concentration. The difference between those concentrations is called the concentration gradient. So we say that diffusion proceeds down the concentration gradient from high concentration to low concentration until eventually the molecules are evenly distributed. Now this is an example, it really has nothing to do with the plasma membrane, but it's showing you how molecules of dye will diffuse throughout a beaker of water. But when we are talking about diffusion, for this chapter, we mean diffusion across the plasma membrane. So when it's diffusion across the plasma membrane, we mean that the substance is crossing through the phospholipids, not through a channel. Well, not if it's just plain diffusion, it's not through a channel or a carrier protein. But diffusion happens with lipid-soluble molecules. They just diffuse directly through the plasma membrane, like it's showing you here. This is a molecule of cholesterol, and it's going to diffuse from wherever its concentration is high to wherever it's low. So if we had more cholesterol outside, then it would diffuse through those phospholipids into the cell, okay? Water and small ions are going to diffuse through a channel protein, and so they're going to diffuse, let's see, water, there's more water outside the cell than inside, so water is going to diffuse into the cell. And it's showing you that large molecules are not going to be able to diffuse unless they are transported by a carrier protein, and we call that something different. It's not just called diffusion. So diffusion can be substances crossing the phospholipid bilayer, and that's going to include lipid-soluble drugs, alcohol, fatty acids, steroids, and gases like oxygen and carbon dioxide. These are all able to just travel through that phospholipid bilayer, okay? And then there's small water-soluble compounds and water itself and ions, meaning they have a charge, that are able to go through, to diffuse through channel proteins. And this is because they are water-soluble, not lipid-soluble. Osmosis is diffusion of water, and it happens because the plasma membrane is selectively permeable. Water is able to cross the plasma membrane a lot more freely than some of the solutes that are dissolved in water. So sometimes instead of the solutes moving from their high concentration to their low concentration, water moves from where the solute concentration is low to where the solute concentration is high. Water is moving toward the high solute concentration. Water follows solute, if you can remember that. Water follows the high solute concentration. And it does that to kind of dilute it so that it can be, so the concentration of the solution can be, can reach equilibrium. So here we have we literally call this a U-tube. We have a U-tube. In this YouTube video, we have a U-tube. And in this U-tube, the solute molecules are shown as purple. And you can see on side A that the concentration of solute molecules is lower. And on side B, the concentration of solute is higher. So here we have a low concentration of solute, and here we have a high concentration of solute. So where is water going to move? Water is going to move toward the high solute concentration. Water follows solute. So the water is going to move this way. Okay? And you see that happen in step 2. You see that the water has moved and the level on side B is higher than it is on side A. And what's going to happen is it's going to do that until there is it's kind of like an equilibrium is reached. There's an equal concentration of solute molecules on both sides. And you're not counting the solute molecules, you're looking at the concentration. So they're about the same concentration on side A as they are side B now. They're about the same distance apart from each other. So we have a similar concentration of solute molecules on each side, so now the water level is going to even out. All right, then we're going to talk about tonicity. Tonicity is referring to the effect of the concentration, the solute concentration on water. So if the solution is isotonic, then it means the solution outside and inside the cells are the same. And there's not going to be a net movement of water into or out of the cell. So the cell isn't going to swell and it's not going to shrink. But if the cell is placed into a hypotonic solution, that means that the solution has a lower solute concentration than the cell does. It's going to cause the cell to take on water. It will swell, possibly burst or lyse. When this happens in red blood cells, we call it hemolysis. When red blood cells swell up and burst due to being placed in a hypotonic solution. So what would be hypotonic to a red blood cell? Red blood cells have a concentration of 0.85% salt or 0.85% saline. This is the concentration a solution would need to be in an IV bag when somebody's given fluids at the hospital. But if we just give them pure distilled water, just pure water with no solutes dissolved in it, then that will be hypotonic to the red blood cell, and it will cause the person's red blood cells to swell up and burst. Now, if we do the opposite, if we give them a solution that is too salty, that's hypertonic. What will happen to red blood cells placed in hypertonic solution is they will shrink, and that is called crenation when it happens in red blood cells. An example of a solution that would be hypertonic would be 5% salt, because inside the red blood cell is only 0.85%, so it's not even 1% salt, but outside it's real salty. So what that's going to cause is water to leave the cell, and the cell's going to shrink. And here's a picture of what happens to a red blood cell placed in an isotonic solution. Nothing, it stays the same size. Placed in a hypotonic solution, it takes on water and it swells up and bursts. And if the red blood cell is placed into a hypertonic solution, it loses water and shrinks. Remember that the situation in picture B is called hemolysis, and the situation in picture C is called crenation. All right, question 8. How would a decrease in the concentration of oxygen in the lungs affect the diffusion of oxygen into the blood? Well, if the concentration of oxygen in the lungs is less, there's going to be less that diffuses into the blood. That's a little bit of, it's one of those questions that makes you wonder, is it really asking something harder than this, or is it really that easy? No, it's really that easy. If the oxygen in the lungs is less, if we take in less oxygen into our lungs and there's a decrease in the concentration of oxygen in our lungs, then of course it's going to diffuse slower into the blood because there's not as much. Number 10, relative to a surrounding hypertonic solution, the cytosol of a red blood cell is, we would call it hypotonic. If the surrounding solution is hypertonic, then the red blood cell fluid is hypotonic. I'm not going to write the whole word out, but I'm just going to put hypo. HYPO. Okay, now carrier-mediated transport is when a carrier protein is required. This uses a membrane protein to move ions or other substances across the plasma membrane. It can be passive, not require any ATP, or it can be active where it requires ATP. Carrier proteins are specific to the substrate that they carry across, like there are glucose carriers, there are sodium carriers, potassium carriers, and many transport only one ion at a time, but some transport more. If a carrier protein transports two solutes at the same time, we call this co-transport. It moves the two substances in the same direction. Counter-transport is when two substances are moved in opposite directions. And we have two examples of carrier-mediated transport, and that is facilitated diffusion and active transport. And we'll talk about those in the next section.